Recording in progress.
Okay. Good. Why is it not showing up? Okay.
Recording in progress.
Oh, no, we cannot do this. Okay. I don't know why it's not showing the screen. So give me a second. My internet connection is unstable, but that's normal. There we go. Okay, some reminders, chapter 16 homework is due today. You will have a quiz this next week. You won't have a quiz. And then the week after, you won't have a quiz either. So I think this is gonna be your last quiz. Okay. No, I can think. So we have a quiz this week. We'll have one more homework that covers chapter 17 and 18. That will be due the week we get back from break, probably. I'll have to figure out what day that is due. Next week, we have a class on Monday. The following week, we have 3 classes. One of them will be a test, either Wednesday or Friday, and then you'll have a final and you'll be done. So the time is moving quickly. There's not a lot left. Questions about schedule stuff? Okay, take a minute, get back into DNA mode. I know you're just singing GTCA, but see if you can name these proteins. This is where we left off on Friday. What are you doing? I don't know what he did there. There we go. These are 4 of the proteins or enzymes that are involved in DNA replication, and that's what we're going to get into the details of that today. Number 1, what is it? The one that unwinds the DNA? DNA helicase. Yes, good. So helicase is unwinding the helix. Number 2, after it's unwound, what comes in? What are these guys called? They keep it from re-annealing and retwisting. They are single strand binding proteins. I don't know why we haven't abbreviated that. Maybe people call them SSBPs. I don't know. Okay, number 3. What keeps it from getting super twisted and tangled? Topoisomerase. And number 4, what's bringing in the RNA bases that are complementary, that starts the process of replication? Primase.
And that, again, that's where we kind of left off. We have our DNA. It is unwound. The hydrogen bonds between the bases are broken. There are single-stranded binding proteins that keep it from rejoining, and topoisomerase is keeping it from overcoiling over here. I don't have like RNAs to bring in to the two sides. So we're going to break down this DNA replication process into two strands. We're going to talk about the two strands separately because it's kind of a different process for the two different strands once it's unwound. But first, we're going to talk about the polymerization process. So when we talk about DNA polymerizing, remember, we're putting together a bunch of monomers. What are the monomers called? Nucleotides. We're going to put a bunch of nucleotides together. We have the template strand, and a new strand is being formed. We have to remember that the DNA strands are what we call anti-parallel, meaning they're running in opposite directions in respect to the way that the sugars are oriented. So these sugars are oriented so that the phosphate groups are connected to the 5' end, and at the 3' end side end corner, I don't know, is the OH group. So we can only add, as we're adding these DNA strands, the bases are going to be brought in, these nucleotides, not just the base. The nucleotide is going to be brought in by the 5th enzyme, which is DNA polymerase. Which makes sense. It's polymerizing, so it's polymerase. It's the enzyme that does it. It's polymerizing DNA. There are 11 different DNA polymerase enzymes that are involved in human eukaryotic DNA replication. E. coli only has 3. We're only going to talk about 2 of them in detail, but that's DNA polymerase 3 and DNA polymerase 1. And we're going to talk about that in that order because that's the way they function in terms of the process, but 3 comes first because just like photosystem 2 and photosystem 1, it's based on when they were discovered. So the enzyme that is bringing in the extra bases, the new nucleotides, is DNA polymerase 3. So DNA polymerase 3, which we call affectionately, of course, Pol 3, is going to bring in the complementary nucleotide. Always adding it to the 3' end. Because that's the only way it can work. It only adds to the 3' end. And that we'll talk about why that is important because it's problematic on one of the strands. This process is a process that also requires some energy. As these are added, some of these are broken off. The phosphate groups are broken off, just like in ATP. This phosphate area, the triphosphates have a lot of energy there. So it's a condensation reaction here to form these, to put these together is a condensation reaction. And it gets its energy from, it's removing these 2 phosphates. It gets its energy from removing those and then breaking this bond. So a hydrolysis of the P to P bond provides the energy for polymerization. So it's a reaction that can kind of provide its own energy that it needs to happen. So the condensation reaction puts them together, but the hydrolysis of the phosphate groups, that P to P bond, will provide the energy that's needed for that bond to be formed. How fast does this happen? How fast would you think this happens? Per second, how many nucleotides per second do you think your body can add nucleotides to a DNA strand? Just guess a number. I'm not expecting you to memorize this. Maybe you did, I don't know. How many nucleotides per second do you think your DNA polymerase can add nucleotides to a template strand? Okay, Noah is overly optimistic. I'm impressed by it, but it's not 100,000 per second. Not quite 200. Okay, a little bit less, you guys heard it. Per second.
72.
Close. It's about 50 nucleotides per second, which I thought was impressive, but apparently is not because it's not 100,000 or 200 or even 100. Okay, 50 nucleotides a second is bringing in. E. coli can do 500 nucleotides a second, so E. coli is a little more impressive than us. I don't know why. I don't know why it's so much faster. But this is happening at a very, very, very rapid rate. So we're gonna talk about it and break it down into teeny little steps, and I want you to keep in mind the whole time, your cells right now are replicating DNA at a rate of 50 nucleotides per second. So it's like 50, 50, 50, 50, 50, 50, and we're gonna take not quite 50 minutes, but almost 50 minutes to talk about how it's doing it. So I think that's impressive.
And again, providing its own energy as it goes. So we've unwound the DNA. We've unwound the DNA. We've kept it from being binding. We're not showing topoisomerase anymore because it gets starting to get messy in here. We've got the RNA primer brought in by which enzyme? Primase. This is the origin of replication. So this is where we're starting. This is our parental strand. So on this side, this is our 5' end, and here's our 3' end. And this is our DNA. Sorry, this is in my way. You can't see it, but. The DNA poly-3 can just bring in bases and add them here because, remember, DNA is directional. So this strand runs 5' to 3', and this strand runs 3' to 5'. So once we've separated them, this top strand, the 3' end is here. So the new strand that we're building is 5' to 3'. So the 3' end is open down here, so we can just keep adding nucleotides. So this strand is what we call the leading strand because replication happens continuously. Because the 3' end is the end that's open, the DNA polymerase just keeps bringing in more bases, more bases, more bases, and it can just keep on going right down this molecule straight. So it's continuous replication or elongation. Because of the direction it's moving, it's adding the bases, the nucleotides to the 3' end. So that free OH group at the end is where it can just keep adding on. And again, DNA polymerase 3 is bringing it in. It's happening at 50 nucleotides a second. It's happening fast. If on this side of the template strand, let's say this is an A, what's the DNA polymerase 3 going to bring in? T. If this is a T, it's going to bring in an, if this is a G, it's going to bring in a, if this is a G, it's going to bring in a, if this is a C, it's going to bring in a, so it's just boom, 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 50 nucleotides a second continuously. The DNA down here is just going to keep on winding. It's going to keep, and the DNA polymerase can just keep going, bringing in the bases. That's the leading strand. That's the easy thing. It's pretty, yes, there's a lot of proteins involved, but it's pretty straightforward. It's not going to be so on the other strand. On the other strand, you see that the 5' end is here. So when we build a new strand, this new strand is going to match up with the 3' end here, moving this way towards the 5' end. And the problem that we have on the second strand is that we can't add bases to a 5' end. DNA polymerase 3 can't add bases to a 5' end. It has to add it to a 3' end. So we can't just stick a DNA polymerase 3, can't just attach here and just move its way down, adding bases as it goes. It has to do it what we call discontinuously. And this is where it gets messy. Okay, so this is what we call the lagging strand. So replication is a little bit slower on the lagging strand because it can't happen continuously. Oops. Today's not going to be the day I don't advance it wrong. The same enzymes are still involved. It's just things are going in a different direction because of the directionality of DNA. So I'm just drawing this up here to show you this is our leading strand. It's still here, but we're focusing on the bottom strand. So this is our origin here. So that's where replication is going to start. But again, because of the way that DNA polymerase moves, it can't attach a base here. Why? Why can't DNA polymerase just bring the base here? It's a 5' and it can't attach to a 5'. It can only attach to a 3'. So we've got a 3' end here, but replication needs to go in this direction because this is where everything is unwinding and unbinding and ready to go. So instead of replication starting directly at the origin, we're going to start with DNA primase down here, adding an RNA primer. That says RNA. What we call downstream. So it's starting with the primer downstream of the origin. So the origin is here. The first primer is going to go here. Now, when we built this primer in this direction, we've got a 3' end here that is free. So now DNA polymerase 3 can start working. So we've got our primer built. Here's our origin. Now DNA polymerase 3 comes in. It only has one L, sorry. It's not a pole, it's a paw. So because it's discontinuous and we're making it in little pieces, those pieces are named after the person who discovered them, whose last name was Okazaki. So DNA polymerase 3 is going to come in and start synthesizing the first fragment. So this is Okazaki fragment 1. Again, moving from the 3' to the 5' direction because it can only add those bases on the 3' end. So it adds bases until we get back to the origin of replication. This is the first fragment. Does that make sense? Okay, only can go in one direction, like a one-way street. So you have to think about like going around the block. I don't know. It's like going around the block to get down to the next block and then around the block again to get to the next block. So once the DNA polymerase 3 hits the next primer at the origin, it's going to disconnect. So it's going to dissociate. So when it hits that next primer, it's going to dissociate and it's going to move back. Not back, it's going to move this direction because this is the direction that replication is happening. So remembering again, the leading strand is up here and replication is moving in this direction. So this DNA polymerase is going to disconnect when it gets here and it's going to go back here to the second primer. So this is the RNA primer for fragment 2. So DNA polymerase is going to make Okazaki fragment 2. So it disconnects here, it comes back here to the next primer, and it starts adding nucleotides down until it gets to this first primer. Then there's going to be another primer. It's going to disconnect and move on to the next thing. You'll notice now we see this enzyme, which is not one that we have mentioned before. This is another DNA polymerase. This is the only other one we're going to talk about. This is DNA polymerase 1. And the job of DNA polymerase 1, can you tell what it's doing here? So this red is the RNA primer. What do you see here? It's replacing that RNA with DNA because we're trying to make DNA. So that RNA primer is there temporarily to get the replication process started. But in the end, we don't want a final strand that has bits of RNA here and there. So DNA polymerase 1 replaces the RNA primer with DNA, with the correct complementary DNA nucleotides. So primase makes the primers to get the process started, then DNA polymerase 1 removes the primers at the end to make sure that we just have DNA. On the leading strand, how many primers are there? 1, because once it starts, it just keeps going continuously. So DNA polymerase 1 is still going to replace that 1 primer, but on the lagging strand, you're going to have primer after primer after primer for every single fragment that you're making. So every time that DNA polymerase disconnects, when it reaches here and goes back to the next primer and then back to the next primer, every single one of those primers has to be replaced with DNA. So DNA polymerase 1 is much more active on the lagging strand than it is on the leading strand. And then you'll notice there's a little gap here. There's one little gap that is left when the primer is removed to the last, this is our 7th one, I think, right? Oh, I didn't numbers that one. I said there were 6. This is our 6th protein or enzyme that we're talking about. The 7th one, the last one that we're talking about, is DNA ligase. That DNA ligase is going to form all the bonds between the DNA fragments. Anyone remember what kind of bonds are between nucleotides? Phosphodiester bonds. Those are between, on the sugar phosphate backbone, it's the phosphodiester bonds. What kind of bonds are between the bases, the rungs of the ladder? Those are hydrogen bonds. So we have hydrogen bonds there that are broken by DNA helicase, and then we have phosphodiester bonds that are made by DNA ligase. So again, all of these enzymes just systematically moving down. So as DNA polymerase moves back to the next primer, DNA polymerase 3 moves down, 1 is going to come in and replace the RNA, and then ligase is going to come in and fill those gaps between the DNA fragments. Is DNA ligase needed on the leading strand? Just that one point where the RNA primer is replaced. So it's only, again, not very active on the leading strand. It's mostly active on the lagging strand. Does that make some sense? Okay, which end of the DNA strand can DNA polymerase 3 attach nucleotides to? Only to the 3' end. Okay, and that, because it can only move in that one direction, that is why we have to have a leading strand and a lagging strand. So the lagging strand is just a little more complicated. This is showing you kind of the overall picture. Again, here's our leading strand. We've got the leading strand template. And then we've got the leading strand being synthesized. And again, it's continuous replication. Everything just moves its way down. The DNA polymerase 3 can just do its job without any interference because it can go in that 3', 5' to 3'. It's just going to add the bases on that 3' end that's open. On this side, it's more complicated because we have so many things that are involved. Oops. You've got primase actively making primer after primer after primer. You've got DNA polymerase 3 adding the bonds, adding the nucleotides at the 3' end. You've got DNA polymerase 1 replacing the RNA primers. And you've got DNA ligase forming the bonds between fragments. Again, at a speed of 50 nucleotides per second. Your cells are doing this right now. And it's not really probably like this. So we don't have a perfect understanding of this process, like we don't have a perfect understanding of any process. How we think it really happens is not so much that the enzymes themselves are moving down the DNA, but the DNA is actually moving through the enzymes. So all these enzymes working together are what we call the DNA replication machine, or the DNA replication complex. And so we think the DNA actually moves through it. Instead of them moving down the DNA is just kind of pulled through it. So this is anchored to the nuclear matrix. So inside the nucleus, this is happening in the nucleus because DNA doesn't leave the nucleus. When we talk about DNA replication, we're in the nucleus. That machinery, these proteins are either anchored to the nuclear envelope itself or to the nuclear matrix. And then the DNA just moves through it itself. And this model, which is the working model that we have right now, it's just not as, it's harder to see than the other one. It's called the trombone model, which I mentioned because I was a trombone player, and so I was excited that it's called the trombone model. Anyone else a trombone player? Low brass. So this, if we get it to work. Oh, I gotta do this. Sorry, I gotta stop sharing. Someday I'll get this too. This is showing you what it looks like more with like the space filling model.
During DNA replication, both strands of the double helix act as templates for the formation of new DNA molecules. Copying occurs at a localized region called the replication fork, which is a Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex. Here, the DNA to be copied enters the complex from the left. One new strand is leaving at the top of frame, and the other new strand is leaving at the bottom. The first step in DNA replication is the separation of the two strands by an enzyme called helicase. This spins the incoming DNA to unravel it at 10,000 RPM in the case of bacterial systems. The separated strands are called 3' and 5', distinguished by the direction in which their component nucleotides join up. The 3' DNA strand, also known as the leading strand, is diverted to a DNA polymerase and is used as a continuous template for the synthesis of the first daughter DNA helix. The other half of the DNA double helix, known as the lagging strand, has the opposite 3' to 5' orientation and consequently requires a more complicated copying mechanism. As it emerges from the helicase, the lagging strand is organized into sections called Okazaki fragments. These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3' orientation. These sections are then effectively synthesized backwards. When the copying is complete, the finished section is released and the next loop is drawn back for replication. Intricate as this mechanism appears, numerous components have been deliberately left out to avoid complete confusion. The exposed strands of single DNA are covered by protective binding proteins, and in some systems, multiple Okazaki fragments may be present.
So there's a couple other videos that are in the PowerPoint that are linked. One of them is Amoeba Sisters. I don't know if you've ever watched Amoeba Sisters. I like Amoeba Sisters videos. There's a link at the top of the Moodle page that has a whole bunch of them, but the one specifically for DNA replication is linked on this slide. So this is on Moodle too if you want. There's two other ones. I honestly didn't watch all of them in completion, but the third one, the yet another video one, is the Amoeba Sisters one. Before we talk about telomerase and telomeres, we have plenty of time. I did want to talk a little bit about DNA proofreading and repair. So we're talking about this happening at a rate of 50 nucleotides per second. And accuracy is really important when we're copying DNA. If we copy DNA wrong and DNA is, the wrong base pairs are there, that can produce the wrong proteins. If it's passed on to the next generation, it can permanently produce a mutation. So it's a problem if you have DNA that is miscopied during the replication process. And it does happen. It's about a 1 in 100,000 mistakes in terms of base pairing. So about 1 out of every 100,000 base pairs, the wrong one is brought in. So instead of like an A pairing with a T, it might be a G is brought in instead. And so 1 in 100,000 doesn't seem like a really big deal, but this is, a lot of these are caught actually by a DNA polymerase enzyme. So there are DNA polymerase enzymes. Remember I said there's 11 of them? There are ones that proofread. So their job is to go through and make sure that everything is matched up correctly and to fix any mistakes. So after the DNA polymerase has proofread, this 1 in 100,000 mistakes might lead to a mistake about 1 in every 10, sorry. No, 1 in every 1 billion. Of your finished DNA, there's a mistake about 1 every 1 billion base pairs in finished DNA. But there are ways to actually correct that after that too. So in the finished DNA, there are other enzymes that are involved that can do what's called mismatch repair. So after the final DNA product is made, there are enzymes that can come through and cut out a mismatch and put in the correct one. There's also, just in general, enzymes that are involved in maintaining and repairing DNA overall, because DNA can be damaged not just in the replication process in terms of making a mistake. It also can be damaged by chemicals. What are some other things that you can think of that you might have heard of that can damage DNA? UV light. If you go outside in the sun and you get sunburned regularly, that can cause permanent mutations in your DNA, damage the DNA to the point that you can get cancer from that. So chemicals can cause it. UV light can cause it. It can happen from just structural physical damage. X-rays, when you get X-rays, when you're exposed to radiation, X-rays, those kinds of things can cause damage to the DNA. And so you can also have, there's a kind of repair called nucleotide excision repair. So there are enzymes that are monitoring the DNA, again, to look for mistakes that didn't come from replication, but that might have happened in other ways, and they are fixed. So there are enzymes called nucleases. I want to take out that word. That excise or cut out the mistake, and then DNA polymerase and ligase will come in and fix it. So there can be mistakes in replication, there can be damage to DNA later, but your cells, again, are still trying to maintain and repair and keep the DNA structure intact, keep those base pair orders intact. Which again, I think is pretty amazing that your cells are able to proofread and later even fix issues that come not from replication errors, but from other physical or chemical stresses that can cause damage to the DNA. Is it perfect? No. Okay, mutations do still happen and do persist to the next generation. We'll talk some about those in the next chapter. And mistakes do happen and people get cancer and things like that because of those mistakes. So it's not a perfect system, but if these systems are not in place, it's problematic. So some people who are more prone to certain kinds of cancer, it's some kind of mistake that's occurred in the system that's supposed to be proofread. So there are people who are more prone to certain kinds of cancer because their bodies don't make the appropriate enzymes and don't proofread the DNA correctly. So they're more prone to, the book I think talks about colon cancer, but there's other kinds of mutations too for other kinds of cancers. So it's not a perfect process, but it's a well-regulated process that your body attempts to do as perfectly as possible. So there's a weird thing that happens with DNA, with linear DNA. So at the E. coli, we're talking about circular DNA, and the
you go all the way around the circle and you're making the complete circle, but when we're talking about our chromosomes, we're talking about linear DNA, so long strands of DNA. And so what happens as we move to the ends of these strands? The end of the lagging strand can't be replicated because there is no 3 prime. So at the end of the lagging strand, there's no 3 prime area, which means that with no 3 prime group, that very last section, DNA polymerase can't come in and replicate that last section. So with each replication, that chromosome gets shorter. Why would that be a problem? Your DNA, every time it replicates, you lose a section. Why would that be a problem? You're losing DNA. DNA has instructions that are needed to keep your body working. So if every time your DNA gets replicated, the lagging strand loses a piece, replication, and it keeps getting shorter and shorter, that would be problematic. So the body copes with that in a couple different ways. So at the end of the chromosome, there are sections called telomeres. And at the end of the telomeres, during those telomeres, there are no actual genes. So the information that's at the end of the chromosome is not needed for coding for proteins because it can't get replicated. It's important that you don't have important information in there. So instead, there are really repetitive sequences. So at the end of each chromosome, there's just a bunch of bases that are tacked on at the end. I don't remember the order of one. There's an example in the book. It's like GTA, CCC, just over and over and over and over and over at the end of the chromosome. So that each time it's replicated, one of those GTA, CCCs isn't replicated, it's okay because there's no information there that is necessary. It's protective. There's also proteins at the end that will prevent the cell from entering apoptosis. What is apoptosis? Programmed cell death. So if your cell sees that something isn't happening right in terms of replication, the instinct is to get rid of that cell because the cell has a problem. Let's get rid of one cell rather than passing on that problem to lots of cells. So these proteins at the end prevent the cell from entering into apoptosis. So you can see here's this last fragment and showing how that part would not be replicated. So that you would end up with a shorter chromosome. So the next time, this is like the parental strand and you would lose another one. So you end up with these shorter and shorter daughter molecules. So we have these safeguards in place to prevent loss of DNA that's necessary. There's also some evidence that shows that this limits the number of times that cells can replicate. So cells have a limited number of replication. So there's research that shows that this is one of the effects of aging, is that your telomeres keep getting shorter and shorter and shorter. And so you can't keep replicating your cells. And that's when they start to show the effects of aging. So my telomeres are shorter than your telomeres. And that's why I'm wrinkly and my hair's turning gray. I don't know. Okay, that's effects of aging. So one of the ways that they're trying to figure out how to reverse effects of aging is by activating enzymes that are called telomerases. So telomerase is an enzyme that will lengthen the telomeres. So this is already active in cells that need infinite replications. So stem cells that need to be able to replicate infinitely have a lot of telomerase that is
active. So even as adults, you have stem cells still in your body. So those would have telomerases active. So one of the areas of research is what are ways that we can make telomerase active on other cells so that other cells are not having telomerases, that are not having telomeres that are shortened and showing those effects of aging. So in stem cells have these telomerase enzymes that will keep those telomeres long so that there's not a limit to the number of replications that can happen. There are some benefits of cells not being able to replicate infinitely. So non-stem cells that have a limit to replications help to limit mutations that can happen. So mutations, again, meaning a mistake in the DNA that is passed on to the next generation. So it becomes permanent. If we limit the number of replications, that limits the amount of mutations that might happen. So it's a way to limit mutations. So there are some pros to that. Some pros to getting old, I guess. You're less prone to mutations. I don't know. Oops. Okay, last thing we have to talk about, and we should get through this without a problem. This is not the same diagram that's in your book. I don't have access to the diagrams that are in your book because Pearson hasn't figured out how to give that to instructors yet. So this is the previous one. But when we talk about all this DNA and the way that we're copying all this DNA, we also have to kind of talk about how it fits into the cell, because if you took the DNA out of one of your cells, all the DNA of your 46 chromosomes and put it end to end, it would be 2 to 3 meters long. So that's longer than I can reach my arms. How big is the cell? 10 micrometers, and you've got 2 to 3 meters of DNA in there. And so the DNA has to be packed carefully to be able to fit in there. There are multiple levels of, remember, chromosome structure. There's multiple levels of chromatin packing. So the DNA double helix itself, if it's like all the way unwound, is about 2 nanometers in diameter. So it's really small. Nanometers are 1,000th of a micrometer. So a micrometer is 1,000th of a millimeter. So we're talking about 1,000,000th of a millimeter. So it's 2 millionths of a millimeter. So it's tiny. So that seems like it's doable to fit it all into a tiny cell. But in order to do that, we form these units called nucleosomes. So nucleosome is about 10 nanometers in diameter. And all it is DNA wrapped around these special proteins called histones. When we talked about cell structure, we talked about the nucleus being full of chromatin. We said that's DNA and proteins together. So this is how those DNA and those proteins work together. So the basic unit of DNA packing is a histone. So a histone, we take, it's 2 wraps of DNA. Bless you. Around 8 histone proteins. And these histones have a lot of positively charged amino acids on the outside. DNA has an overall negative charge. So that helps that ionic attraction, not ionic, that electrical attraction helps to keep them tightly wrapped. Then they have these tails that stick out, these histone tails that stick out, and those are things that can be modified when genes are expressed, which we'll talk about in chapter 18. But when you stretch this all out, it looks like beads on a string. So we think of the nucleosome as beads on a string. So each one of these beads is one of these histone packages with the DNA wrapped around it. And the DNA in between is called the linker DNA. Okay, then those fibers are twisted around themselves and looped. And again, this is where it gets a little bit different from your book, because our understanding of DNA packing has changed a little bit between the 2 editions. So the next layer is kind of this looping, where these histones are gathered into a chain with the linker DNA sticking out like loops. This is the kind of DNA that you see in interphase. So remember when you looked at interphase under the microscope, you could kind of be like, oh, look, there's the nucleus because it's darker, but you couldn't really see the specific structures of DNA. It didn't look stringy because it was more in this relaxed format. The condensation of DNA occurs as we move into the stages of mitosis, as these loops start to loop upon themselves and they make, I'm not an artist. You can see why I didn't major in art. They make more of like a flowery kind of structure where you've got scaffolding proteins on the inside. And this is what you would see more as we get into prophase and prometaphase. As the DNA starts to condense, and then those can actually wind upon themselves too. And this is what we get to the point when we get to the metaphase chromosome, which is the most condensed. It's the shortest chromosome. So that DNA just keeps packing and packing and twisting upon itself, twisting around itself until you get to this most condensed metaphase chromosome. There's two types. We kind of categorize this chromatin. I didn't mean to do that. Erg. Okay, I cannot minimize that while I'm recording. I don't know what happened there. We divide the chromatin into two categories. We have euchromatin. This is in its looser form when it's not as condensed. When it's euchromatin, it's accessible for things like replication in the S phase of interphase, which is when that whole thing that we just talked about happened, and for transcription and translation, which we'll talk about in the next chapter. So once it's super condensed like it is in metaphase, it's not very accessible for things to happen to it. So in this case, when it's heterochromatin, it's condensed, and the genes that are in it are not expressed at that time. Because it's not accessible to the machinery. I think. Sorry. So the form that the DNA is in can affect how it can be used. Transcription and translation. You need to transcribe. My voice needs to be transcribed automatically for you, or you have to use translation to figure out what it's saying when I'm actually writing. And this picture at the bottom is just showing you a nucleus that's actually in prophase, but the DNA is still organized into chromosomes. So they actually tagged the chromosomes with different colors. So you can see that there are different colors because the different chromosomes are still organized, even though they don't look like the X-shaped chromosomes or the linear chromosomes before replication. Okay, that's chapter 16. We will start chapter 17 on Wednesday, which is transcription.